So happy Friday the 13th to everybody who's here. Yeah, congratulations or commiserations for actually getting anybody slide over on the way in today. Anybody have more than a foot of snow at their house? That'd be me. So just in case you had some ideas about whether having class today was a good idea or not, you can email these people. Let them know how you feel about it. So that being said, hopefully everybody did have a chance to find Wednesday's lecture. Yes, posted on YouTube. Wasn't my best effort, but and hopefully you don't hear my daughter too much in the background while I'm recording it. So that is material which will be on exams when those come along. And so any questions that come up for that, please let me know. I think I may have mentioned in that recording to send questions to D2L. That's not a good idea. Just send questions to my normal email address, kstedman@pdx.edu, and I will forward it to these people if I think it's appropriate. So you may have also noticed there were a couple of clicker questions in that lecture. We are going to have clicker questions today. They don't count towards your grade. They won't count until next week. So for the number of people who aren't here, that will hopefully make their lives a little happier. But today we are going to have a couple of clicker questions because they're just as important for me, I think, as they are for you. And our first one for today. Yeah, sure.
If we have more cancellations due to weather, how are we going to address point situations?
We will deal, sorry, I'll just repeat this for people who aren't here because there are a lot of them who aren't. If we have other cancellations, how are we going to deal with the points relative to the clicker questions? I had another question about this that somebody sent me online. I think I posted to D2L in discussion groups. Basically what I do is I normalize all the clickers to the highest scores. And so if we don't have class, we're not going to have clickers, but that means there's going to be a 0 for everybody for that class. So yeah, the normalization, fortunately, I didn't think about it that way, but it ends up working out in our favor as far as that's concerned. So yeah, other questions about clickers before we actually go here? What's the frequency? It's AA. So yeah, sorry, I try and read these things too. Almost all polymerization reactions in biology are blank reactions. Acid-base catalysis, condensation, hydrolysis, dehydrogenation, or reduction. And think yourself lucky I don't usually lecture that speed. And a number of people have asked, what level of chemistry do I expect for this class? This is about that level of chemistry. So I'm not going to ask you about Diels-Alder reactions or ask you to push electrons around. That's what OChem is for. Oop. If we can keep this projected, that would probably help too. Sorry about that. Oh, back one. How could I? There you go. So I'm not sure how many of you have your clickers with you today, but there are 130 some people in this class at this point, and a whole 37 seem to be here today. Yeah, again, congratulations to those of you who actually did make it. The question here again, acid-base catalysis, condensation, hydrolysis, dehydrogenation, or reduction. 99% of the reactions we're going to talk about have to do with water in this case. So that brings us to basically only B or C. And hydrolysis is doing what? Is it adding water? So if you're adding water to bonds, what's that going to do? It's going to break them apart. So that only leaves you with condensation. So condensation is the correct answer. Yay for the 46% of you who are here. But again, it's not even going to count. So our next question, you are told that the dissociation constant for a protein-DNA interaction is nanomolar. What does that mean? It means there's a very tight binding between the protein and DNA. There's a weak binding between the protein and DNA. There's a very little non-bound protein. There's a lot of non-bound protein, or A and C. feel free to chat about this. This is not like, you know, nasty exam stuff. So I know there's nobody here, so it's hard to talk to anyone. But yeah, these are kinds of things you'll see in the literature all the time. Is what kind of dissociation constants. This is why I spent a while talking about KDs in the lecture on Wednesday. And I either did a good job of talking about it, or everyone knows this already, one or the other here. So yes, the whole idea of the dissociation constant, it's looking at the two different rates of binding and rates of dissociation. And so if you look at a comparison of those two rates, if it's nanomolar, that means it's a really small amount which is not associated. And so if a small amount is not associated, that means that there's going to be very little non-bound protein. So C, and it also means that there's tight binding. The reason there's very little non-bound is that there's very tight binding. Do people get this? No, yes, maybe? Of the 36 of you who are here, and yet at least 19 of you seem to have it. But this is when we talk about nucleic acid interaction proteins, which we're going to do a lot of through the rest of the course. Almost all of those are going to have nanomolar dissociation constants. So they're going to bind really tightly to DNA. And basically what that means is they're always going to be bound to DNA as far as we're concerned under the conditions that we're talking about. But we are chemistry, and so it's always going to be in equilibrium. Just in this particular case, the equilibrium is really shifted, again, in these cases of nanomolar dissociation constants, relative to your DNA, and so it'll always be associated in that particular case. So, and quick review on the other stuff that I didn't have in these clicker questions. One of the nice things about clicker questions is it does give me an opportunity to review a little bit. Again, atoms in biology that we care about, 5 or 6. Most of the rest of them we can ignore, at least as far as this level of molecular biology is concerned. Strong interactions, I'll start over here on the other side. Basically, this is all about covalent bonds. And how do you form covalent bonds? You have to have some kind of, what kind of reaction? Usually polymerization, condensation reactions. And those are energetically non-favorable. So in order to get these non-favorable reactions to take place, you need to have some kind of coupling, and this is almost always to some high energy intermediate. Very often it's the precursor that you're actually using for that reaction. Adenosine triphosphate is a classic example of that, where you then activate your intermediate in such a way that your condensation actually is going to be, when coupled to this other reaction, a spontaneous reaction, one that gives you a negative delta G. So once you've made all of these things, which we'll talk about a little bit more later, when we talk about polymerization processes and DNA polymerization and RNA polymerization, but the interactions between these things all have to do with weak interactions. Yeah?
You said in the previous lecture that you don't really like to think about them as hydrophobic interactions, and I'm kind of unclear why were you thinking of them more as like the two molecules that aren't hydrophilic are interacting?
Yeah, so this is kind of a question. Obviously, you listened to the lecture. Great, thank you. The question is, why do I not like the term hydrophobic interactions? I probably misspoke a little bit there. A lot of people talk about hydrophobic bonds, which is totally and utterly wrong. And the hydrophobic interaction, as far as I'm concerned, is really more about the hydrogen bonding that forms between water molecules. So it's more of an exclusion. So it's really it's really hydrogen bonding, which leads to hydrophobic interactions. And so maybe I didn't explain that well enough. I'm not sure.
To the two molecules that are hydrophobic interacting, it's more about the water.
It's much more about the water squishing them together. Because if you happen to have these in an organic solvent, for instance, not in water, they would not be interacting in any way, shape, or form. So yeah, well that's okay. We've done the hydrophobic interactions, good here. But yeah, the other weak interactions, these are the ones which are very important for molecular biology. Again, I love to use the DNA protein interactions, which we'll talk a lot about because they're very important. And those interactions have a couple of things that they need. One is specificity, so you've got to have very specific interactions, and we'll talk about how some of that happens a little bit later on, but that's due to multiple weak interactions. So many, many, many weak interactions is really about how molecular biology, once you've made the macromolecules, is really about. So it's the van der Waals interactions, hydrogen bonding, and ionic interactions, and multiple of those which will give you specificity, but also reversibility. If you had all really, really strong interactions all the time, then life wouldn't really happen because you need to have some kind of dynamics in the system to be able to do this. So any questions about the chemistry stuff from Wednesday? No. Okay, so let's move on and talk a little bit about proteins. This is kind of a nice lead-in to the protein section. Again, this is a whole chapter in the textbook that we cover in one lecture, but don't worry, we'll spend a lot more time on some of the other ones. But this is basically to give you the background material, and hopefully most of this is review. Yes, most of review. People have heard a lot about this before. Good. So we'll talk a little bit about the amino acids. These are the monomers which are involved in putting the proteins together. These amino acids have molecular biology polarity to them. They've got a front end and a back end, the amino terminus and the C terminus. And this is very important, just like I talked about nucleic acids in lecture number 1. Having that orientation is very important. How do amino acids get put together? Through peptide bonds. Once you have this stretch of amino acids that are all linked together from amino terminus to C terminus, through these peptide bond interactions, then those amino acids can orient themselves relative to each other. And that gives you, as long as you're talking about backbone interactions, and we'll look at the backbone in just a second, the so-called alpha carbon interactions, those then are going to give you secondary structures. Your protein folding is usually after you have secondary structures, and the secondary structures come together, and that will give you a domain. And a domain is a separate part of your protein that has a separate structure, which gives a separate. Thank you. No, there aren't very many of you today, so it's a little more challenging. Some proteins just have a single domain and have a single function, but a very large number of proteins, particularly those that are present in our genome, have multiple domains and then multiple functions. Sometimes you'll have multiple individual polypeptide chains, so it's got C terminus and N terminus. That would be one polypeptide chain. You got 2 of those, or in many cases a lot more, that come together. Those are multimers, and as we'll see as we move on through the course, the vast majority of proteins that are involved in molecular biology are multimers. They've got multiple different polypeptide chains encoded by different genes that come together in many cases, but also cases it's just one gene and then 2 molecules are coming together as far as that's concerned. Protein families, we talked about this last time in terms of how they're related to each other. We'll talk more today about the structures of these different proteins and how they're related to each other. And of course, if you've got a particular structure, it's going to have that particular function as well. And if we have time at the end, we'll talk a little bit about modeling and allostery. Allostery we talk about here is minimal. Allostery, as far as biochemistry is concerned, very important, and hopefully they'll be covered in your biochemistry course. So let's start by looking at the building blocks, how you put these proteins together. As I mentioned, these are the amino acids. Amino acids, by definition, have an amino group at one end. Here we've got our NH3 and a carboxylic acid group at the other end. In the middle, they have one carbon atom. That's the so-called alpha carbon. And then, of course, what makes all the different amino acids different from each other is what's attached to that alpha carbon, or the side chain. Very often you'll hear me talking about individual different side chains as well. So these are just 4 different examples: methionine, aspartic acid, leucine, and tyrosine. All of these amino acids can come together to make, in this case, a short peptide. This is 4 amino acids put together, and they're always linked from carboxy to amino, carboxy to amino, carboxy to amino, etc. So just like you have in the monomers an amino terminus and a carboxy terminus, in a polymer you have an amino terminus and a carboxy terminus. And what makes these proteins, of course, interesting, i.e. giving them structure and then function, is these different amino acid side chains on the outside, just shown by these sort of cones here at the bottom. Drawing all of this out is kind of a pain, and so almost all the time you'll talk about the sequence as we have down here at the bottom, where we've got just the individual amino acids drawn out. Even easier is to look at them in the single letter code. I don't expect you to remember the single letter code, but it's a very useful one. In fact, that is what we see here. Here are single letters, quite easy to remember, except for some of the weird ones like this one, lysine, which is K, glutamate, E, aspartate is D, leucine and isoleucine, proline are all pretty straightforward. Couple of different things. Now, I don't expect you to remember all the different amino acids, but I do expect you to remember a couple of really important ones. And those are the ones basically at the top on the left here. Aspartate, glutamate, arginine, and lysine. Why? Because they have a stable charge at neutral pH, which is what's present inside the cell. Exactly right. So they do have stable charges. Histidine, on the other hand, is also listed here, but that charge is very dependent on the local structure, which is there. So sometimes it's going to be stably charged, sometimes it's not. The pKa, which if you like to, we can get into ad nauseum, but I don't consider it to be really important for this class, is quite close to neutrality. And so that sometimes will be charged and sometimes won't be. There are also a lot of these so-called non-polar amino acids, and now we're getting back to talking about chemical polarity as opposed to the polarity of molecular biology here. And that just has to do with the charge or separation of charge. And mostly it's going to be in terms of interacting with water. So any of these polar amino acids or the charged, stably charged amino acids are going to be interacting with water, which means they're going to be the outside of your proteins for the most part. And all the non-polar amino acids are going to be the inside, with, of course, lots and lots of exceptions about that. This is what I expect you to know in terms of the individual amino acids. I will try again to write them out at least as far as their three-letter abbreviations, but it's always good to know E, D, K, and R. Lysine and arginine have these amino groups on their side chains that under neutral pH are almost always charged because they have an extra hydrogen atom that's associated with them. On the other hand, glutamate and aspartate almost always have negative charges because they've given up this hydrogen atom. And these are the basic or acidic side chains. And again, this has to do with the charge. We'll mostly notice, in fact, the positively charged amino acids down here. Why do you think that is? Give you a hint. He keeps talking about nucleic acid interaction proteins. Nucleic acids have what kind of charge? Negative. So positive, negative interactions. So ionic interactions, very important as far as these are concerned. We'll talk a lot about lysines and arginines as we move on through the rest of the course here. So those are the only ones I expect you to remember. The rest of them, fascinating, important, critical. Take Dr. Bartlett's protein structure class next year and hear a lot more about them. In fact, he's teaching, I think, right at the same time as this. So if any of you are taking it simultaneously, you're more schizophrenic than I am. So now a little bit more about protein structures. Mentioned this before. We'll probably be beating this over the head a little bit too much. But primary sequences, secondary sequences, primary structures, secondary structures, tertiary structures, quaternary structures. Primary structure is just the list of amino acids from N-terminus to C-terminus, or the sequence of your amino acids. So That's your lysine, arginine, leucine, glycine, etc. Just that stretch of amino acids, regular molecular biology polarity, going from N-terminus to C-terminus. Those then individual amino acids are going to interact with each other to form secondary structures. And those secondary structures, we'll look at them in much more detail. The two that we care about and the two by far most common are the beta-pleated sheet and alpha helix. Those have to do with interactions that can really happen in any peptide chain, with the exception of proline because it makes kinks. But that's because all of these guys have the amino terminus and the carboxy terminus. And so they can interact with each other, each individual amino acid, in the absence of a particular side chain. And what we'll see when we look at the individual side chains, one of the main things about secondary structures is it gets the side chains away from where your peptide bonds are. And so it's sticking them out. Alpha helices stick amino acids out perpendicular to the alpha helix. Beta-pleated sheets, up and down relative to that as well. You take those secondary structures, those interact with each other. Now we're interacting via the side chains, and that gives you your tertiary or three-dimensional structure. This, again, is just one peptide chain here. It's one molecule. One N-terminus, one C-terminus hidden in here somewhere. I'm not quite sure where it is. Then if you have multiple polypeptide chains, multiple molecules coming together, that's going to be your quaternary structure. How do you get from your primary structure over here to the quaternary structure here? It's all about the individual amino acids here, the individual amino acids on your side chain. If you just have a stretch of amino acids, 90+% of the time, that's going to give you one and only one structure at the end. Full tertiary, or if it's interacting with other proteins, quaternary structures. And again, these are all interacting now with each other, the different side chains, through those weak interactions that we talked about before, with van der Waals interactions, ionic interactions, and hydrogen bonding interactions. People look at protein structures in lots of different ways. The simplest way is just to trace along where the peptide bonds are. And that's shown here. Very often, people have this as a color scale, going from red to blue or red to violet, starting at one end, going around to the other. That's not a very useful way to look at proteins, because what are they interacting with each other? It's going to be through their various different side chains. Probably the best way of looking at protein structures is down here. These are now your van der Waals interactions, the position in each of those atoms where you start to have repulsive activity if he gets too close. So this is what other macromolecules will see. Unfortunately, it's kind of hard to see your way through this. If you're looking at individual amino acids on the outside, it's okay. But to understand the whole structure starts to be a little bit difficult. So what we usually use is this sort of cartoon model here, where you have a helix describing the alpha helix, and each of these bands here describing the beta strands fitting together here into a beta sheet. In this case, it's a three-stranded beta sheet. But all of these are representation of the same protein. So it's just a slightly different way of looking at it. This is the way we're mostly going to be looking at them. Doesn't, however, give you side chain interaction information. Down here, this is your space filling. You can use this structure too, but trying to find things in here is kind of a real pain. Some of you might notice, if you look really carefully, that there's a, phenylalanine ring right there. It's hard to tell if it's a phenylalanine or a tyrosine, if there's an extra piece hanging off of it at the end. But those are the kinds of structures we'll be looking at. So let's talk a little bit more about the secondary structures. Talking about secondary structures in Portland, Oregon is a wonderful place to do that. Why? Because Linus Pauling was the one who came up with them. Where's Linus Pauling from? Portland. So just over the river on the other side here. So he and Corey came up with these predictions, actually, for structures. just based on what they knew about the individual amino acids, and then a small amount of data on proteins that were either silk or hair, and then came up with these really amazing, basically, Pauling is credited with coming up with a hydrogen bond. And a lot of that is because of looking at these secondary structures. So, probably one of the more brilliant scientists ever, as far as I'm concerned. So, basically, to put these things together, what you have are hydrogen bonds which form between your backbone atoms in your polypeptide chain. So this oxygen right here, if you're just an amino acid, that's part of your carboxylic acid that would be right at the end of any of your individual amino acids. And that interacts with a hydrogen right here, which is part of that amino group. And so it's the carboxyl aminos that are interacting with each other in order to form hydrogen bonds. And as long as those are doing that, those atoms are interacting with each other in the same local area, a couple of amino acids apart, that's going to give you one of these helices, and it's a little hard to see on here, but all of the R groups, these are your side chains, are all sticking out around the outside. So they're free to interact with another part of the protein in terms of your tertiary structure, or they could be interacting with a completely different protein or a nucleic acid, etc. So this is your classic alpha helix. There's going to be slight modifications to each of them. One of the things that was found quite early on in terms of talking about DNA structure is it turns out that alpha helices fit perfectly into major grooves of DNA. I wonder why that is. Wonders of evolution. And also, these are classic helical structures. We're going to talk a lot about helices in this class. I have a collection of various different helices here. If people are interested in looking at left-handed helices, right-handed helices, combinations of left and right-handed helices. So I also kind of like to think of this as a various different armed helix. Is this a right-handed helix or a left-handed helix? Right. And the way that I would tell this is I would grab an arm and sort of look at, okay, if I wrap my arm around this, it wraps around that way going up. If I try and wrap my left around that, that's not the appropriate way. So it's the right-handed helix here. Yeah. I see people waving their thumbs and fingers here. But one of the things that I asked people to do a couple of years ago, and then I got this whole collection of them, was to bring me left-handed helices, examples thereof, and that's what all this collection is. How many people know what this is? I know, John, you know what this is. But yeah, I'm getting old. What can I say? It was actually hard to find some of these things. So alpha helices is the one common form of secondary structure. The other one, also discovered, or at least proposed by Linus Pauling, was the beta-pleated sheet. And now it's basically exactly the same thing. You've got your hydrogen atoms from each of the amino groups in your polypeptide chain and the oxygens from your carboxylic group. But now instead of local amino acids, they're reaching across to amino acids that are further away in your primary sequence. And sometimes actually even on different molecules. And we'll see that in just a couple of minutes here. But basically, it's those interactions that can happen with any set of amino acid side chains which are now giving you this structure in a very analogous way to your alpha helix, where you're sticking the amino acid side chains out to the outside, and now you're sticking them up and down relative to each other. So moving them away from the backbone so they can interact with other different things. You can have either anti-parallel here. Now, this anti-parallel has to do with the molecular biology polarity, N to C. So N to C in one direction, C to N in the other, N to C in the opposite one. But you can also have all of these in parallel to each other. So it's not like nucleic acids, where you always have to be in opposite molecular biology polarity to bind. Here you can be parallel or anti-parallel relative to each other. So does that make sense? Yes, no, maybe? Are you ready to answer a clicker question so I can take a breath? Okay, so here, protein secondary structures are due to van der Waals interactions between side chains, hydrogen bonds between side chains, van der Waals interactions between backbone atoms, hydrogen bonds between backbone atoms, hydrophobic interactions, or water interactions by themselves. Knew I forgot something in my water bottle. This one's not mine. Got 7 more people who've come in. All right, 8. More. No, this is not like the U.S. elections where you can vote multiple times. No. It is the last one of your elections which ends up counting. So up to maybe a third of the people who are here. So what are the interactions which are giving you the protein secondary structure? They are, yes, hydrogen bonds. We seem to be pretty agreed between that. But are they between the side chains or between the backbone atoms? It's the backbone atoms. The side chains are supposed to be interacting with other molecules. That's the whole point of the secondary structure. So you're sticking the side chains away. So they're not going to be interacting with each other, at least not in the local environment. So what happens? So let me actually choose the correct answer here. I always forget to do this. Please remind me when this actually does start counting. Okay, so yeah, so now we've got our secondary structures. How do they then make a domain a real tertiary structure? It's that tertiary structure that you have to have in terms of getting activity. Just a single secondary structure, with few exceptions that we'll talk about, really doesn't have activity by itself. And it also, and probably much more importantly, is not stable by itself. You have to have multiple secondary structures come together in order to give you something which is stable. So how do they interact with each other? It's the side chains. It's all of those weak interactions that we've talked about before. Ionic bond interactions between stably charged acidic side chains, basic side chains. Hydrogen bonds that form between these different van der Waals interactions that form between them. And then all the water molecules associating with each other on the outside, wanting to maximize the number of hydrogen bonds that are there, thereby squishing together the nonpolar amino acids, which almost always end up in the middle of your protein down here. So that process, going from your primary sequence, forming secondary structures, that usually happens very fast. If you look at taking a polypeptide chain, particularly if it's being made on the ribosome, that then will make secondary structures very quickly. It's getting those secondary structures together into your tertiary structure, which takes a lot longer. And that's the whole so-called protein folding process. It would be really, really amazing if we could take that stretch of amino acids just by looking at it. And since we know the genetic code, if we've got nucleic acid sequence, we can know what the amino acid sequence is, and then predict what that stretch of amino acids is going to do. The way to do that would be able to predict the structure. So go from your primary sequence of amino acids to the tertiary structure, which would tell you something about the activity. People have been working on this for about 40 years and still have not come up with a good answer to this problem. The problem is, they mentioned in the textbook, is there are way too many different ways that these things can interact with each other. And, as we'll see a little bit later on, the actual active site, or what's actually doing the job of your protein, is usually only very few amino acids in your actual whole protein. So most of the protein is actually just serving to hold the few side chains that you need for activity into one place. So it really makes this whole folding problem, as people call it, extremely difficult. Did want to spend a little bit more time talking about helices. Why are helices so wonderful? Because they don't stick side chains out on the outside, but probably much more importantly is helices are formed when you have almost identical interactions with each other. Each of the monomers can then interact with its neighbor in an almost identical fashion, and you can end up with one of these really nice long structures here. There's no particular reason that you should have left-handed versus right-handed helices. It just has to do with the chemical bond angles that you have in peptide chains, that you end up with your normal alpha helical form. You actually can get some of these different forms. You also can get different forms in DNA. But just these interactions of helices, and again, if you're not clear on helices or handedness, I saw people holding up your fingers and that kind of thing, come down and take a look at these models, because I think it makes it really quite straightforward. But just visualizing, I have a lot of trouble seeing it in this 2 dimensions here. I find it a lot easier looking in 3 dimensions as far as that's concerned. Why do we care so much about helices? Yes, they've got all these nice things that stick out on the outside. But one of the things that happens very, very frequently with these alpha helices is you have 2 alpha helices, often from different molecules, that then interact with each other. And this is best called a coiled coil, where you literally have 2 alpha helices that wrap around each other. Many more people will talk about these as so-called leucine zippers. Why is it called a leucine zipper? Leucines are one of those non-polar amino acids. So they're being stuck on the outside of your alpha helix, right? So what are water interactions going to want to do? They want to try and push those non-polar amino acids towards each other. So you have an alpha helix with leucines sticking out around it, particularly if you have leucines every 7 amino acids. I forgot to mention it's actually every 7 amino acids as you go round. You end up in exactly the same place on one of these alpha helices. So you've got leucines or some large non-polar amino acid residues sticking out on the outside. When I say residue, I just mean side chains. Sorry, it's this. I'll try to talk about side chains before, but residue is if you break stuff down. It's old school biochemistry. But if you have these leucine side chains, every 7 amino acids, they're going to interact with each other almost in a way which is pretty similar to a zipper. A better way, again, of talking about this is a coiled coil, literally where you have 2 alpha helices wrapping around each other. But if you have leucines, is a classic way of looking at that. And this is, in fact, a structure here right in the middle, where you can see each of the leucines shown in red now interacting with each other. And again, the reason they're interacting with each other is because the water is basically excluding everything else. This kind of dimerization motif is very common, particularly in nucleic acid binding proteins. There's a similar kind of coiled coil that happens in collagen, a lot of connective tissue. Turns out to be not alpha helices, but again, these coils wrapping around each other. This can give you really quite stable interactions between the 2. So this is another way of looking at that here. If you just count your way from N-terminus to C-terminus along an alpha helix, just lettering, A for your first amino acid, B, C, D, E, etc., all the way along here, you'll notice that all of the E's are on the same side. The A's are on the same side, so if those have your large hydrophobic amino acid side chains, again, very often leucine, those can now interact with each other. So that's just individual secondary structures. Usually just the coiled coil by itself is not actually stable. It has to be attached to something else. And that something else is usually going to be now a domain. Now, domains are a separately folded piece of your polypeptide chain. It has a 3-dimensional structure. That 3-dimensional structure is usually stable. We'll talk about some non-stable structures a little bit later on. This part of a protein, this is a classic example of one of these proteins here. Now, in ribbon diagrams, this particular one has 3 or maybe 4 if you count this extra one out here, different domains of your protein. This one has one particular structure, this is another structure, this is another structure, and not surprisingly, they have different functions relative to each other. So we'll talk a lot about nucleic acid binding proteins. It'll be a transcriptional activator. It'll have one domain that interacts with the DNA and a different domain that's going to interact with parts of the transcription machinery, for instance. So multiple domain proteins, particularly true in some of the larger eukaryotes, not higher eukaryotes, as my yeast biology friends like to remind me all the time. But in the larger eukaryotes, very often you've got multiple domain proteins. And a lot of times, these domains, I should say, get mixed and matched. So you'll have one protein that has this particular domain in it, but another protein that's got multiple different domains from different proteins. And this seems to have happened in evolutionary processes, and we'll talk more about that when we talk about genome structure a little bit later on. But mixing and matching different domains with each other on one particular protein seems to be able to give you lots of different activities, but basically using the same sort of modules. So moving these different modules around allows you to get lots of different kinds of activity. What do these domains look like? They can be all alpha helix, all beta sheet, some combination of the two. This is just basically to show that there are lots of different ways that these things can be interacting with each other. The so-called 4-helix bundle here is a really classic structure in a lot of the work that's been done in trying to figure out how you can get a particular stretch of amino acids to form a particular structure, and some of the protein folding predictions have been done in this really simple form, and you do find these quite often. Beta strands here now all parallel to each other with these alpha helices in between them. Also what's called a beta barrel structure. So this is basically a beta sheet which is wrapped all the way around, giving you a continuous structure. Many proteins have this particular structure in them as well. So now I wanted to talk about how we can predict protein function from looking at sequence and trying to think about how these things are associated with each other. We can't go from amino acid sequence to predicting the structure and predicting the function. What we can do, however, is if there is a structure for one stretch of amino acids, and we can look at similar amino acids and say, oh, if we compare these sequences to each other, we know what this structure is. Now we have this sequence. These sequences are similar. Are they likely to have the same structure? They have the same structure, same function, or at least similar function. And it turns out that this is a really powerful process and actually works extremely well. Pretty much any time when you have amino acid sequences, primary structure, just N to C terminus, which have 30% identical amino acids across whatever stretch of amino acids that is, that's going to give you 99.9% of the time the same structure. So if you happen to have a structure of, say, in this case, chymotrypsin, and you have a new sequence that's come from your favorite organism that you found in some deep-sea hydrothermal vent that you've been sequencing, you can say, hey, I've got this stretch of amino acids which looks a lot like this stretch of amino acids that they already have a structure and function for. So you can make predictions. Anytime you've got your amino acids sequence which is higher than 30%, pretty good indication that you'll have something at least with very similar structure. One of the things that they mention here, in fact, these are two sequences. Just the green here represent the identical amino acids. This is about 30% identical. Lots of them which are not. So there's gray loops here on the outside. But elastase and chymotrypsin are two proteases which have very similar kinds of activities, slightly different amino acids that they're going to be interacting with. But 30% identical, you can really make a prediction this is likely to be a protease. On the other hand, there also are some examples of sequences that are very different relative to each other. So this is a case of a DNA binding protein, yeast and Drosophila. These are extremely different. It's a little bit hard to see up here, but very few of these amino acids. So the line between the two, so these phenylalanines over here, glutamates there, leucine here, they're only about 17% identical, but it turns out their structures are almost identical to each other. So if it's more than 30%, you know the structures are going to be very similar. Less than that, they might be, they might not be. And in those cases, you actually have to do a little bit more work to figure out what's going on there. Yeah?
Is that just because the majority of the amino acids are connecting to each other or aren't bonding with each other?
So maybe I'm not sure I completely understand your question, but I'll try and paraphrase it here. Is the reason that you can have the same structures with very different sequences because a lot of the amino acids are not involved in the real activity? So that is probably true to some extent, but if you look at these particular structures, so these are the two structures here of this yeast protein and the Drosophila protein overlaid with each other, and even though they're only 17% identical amino acids, with the exception of this little loop here, they're basically identical in structure. And that's for the whole thing. The business end of this protein is a DNA binding protein, so it turns out it's just one of those helices, which is important for the function. So it's not just that. Yeah?
So the ones that share the amino acid sequence greater than 30%,
Yeah.
is it believed that all that's been inherited, that sequence has been inherited between the two, or is it just kind of
Random or convergence or something like that? Yeah, so that's, in fact, a great question, which leads me to the second part on this slide. So the question was, why do you have this kind of amino acid similarity? Is it due to some kind of common ancestry, or is it some kind of convergent evolution which has taken place? And the answer to that is it's probably, and again, there are always exceptions. This is biology, which is annoying. But yes, I would say 90+% of the time, most of the time, it is going to be some kind of common ancestry. And you can see that through a lot of these different protein families that we talked about before. So almost always is going to be common ancestry. There are some examples of convergence where you do end up with clearly very different things coming together to make really similar kinds of proteins. But that's pretty rare, other than with viruses, which are the coolest things ever, but we'll talk about that next term in virology, maybe. But yeah, most of the time it's going to be a protein family, and so these would be nice, conserved, probably homologs. They've been inherited from some common ancestor. Lots of different kinds of proteins that can come together. The basic message that I wanted to get out of this image is that incredibly variable in terms of size. Some of the smallest proteins, like insulin here, are pretty tiny. Single domain protein here has that particular activity. Some proteins are ginormous, multiple subunits here interacting with each other. This is the glutamine synthetase. It's got 6 different polypeptides that are associated with it. Antibodies, there are 4 different polypeptides, this guy here, and then these 2 guys here down at the bottom. Just for scale, RNA never looks like this because it's always base paired with each other, but to show the relative scale. And double-stranded DNA, B-form DNA, also pretty similar in terms of these scales. If you look at a pretty typical lipid bilayer on a membrane of a protein, you can see that a lot of these proteins are about the same scale as these lipid bilayers. And they spend a while talking about transport proteins. That's more for a cell biology kind of process here. So scales are pretty similar with a lot of these macromolecules. So you can think about interactions between proteins and membranes, proteins and nucleic acid. They're going to be about the same kind of scale, which when we talk about this later, you'll see some of the cartoons in the textbook, they're totally out of scale. So they've got the nucleic acids as being really big and the proteins as being really small. We'll talk more about that when we actually get there. A lot of these are interacting, as I mentioned before, as multimers. All the multimer means you've got multiple molecules that are interacting with each other. I mentioned where you can have beta strands interacting with beta strands on a completely different molecule. That's the example right here down at the bottom. This is a regulatory protein, DNA binding protein, from a virus, of course, the coolest kinds of things that are out there. But the main thing here is we've got a dimer, so 2 macromolecules that are going to be associated with each other. And it turns out this is absolutely required for activity. If you change any of these interactions here, this molecule doesn't work anymore. Another nice example of a multimeric protein is this one, neuraminidase. Anyone know where neuraminidase comes from? Everybody get their flu shot? Part of the flu shot is the neuraminidase. So one of the proteins on the outside of the influenza molecule. This particular one is a tetramer, but it's not so important that there are 4 of these that come together. But what is important is it turns out that the active site where this enzymatic activity is actually between these 2 monomers. So it turns out that many of these multimers actually are bringing together amino acids that have to be part of the active site. So actually only going to be active when they're multimeric proteins. So they have to come together in order to form these particular structures. Yeah?
Are all tetramers that way, where you have to have 2 of these amino acids together in order to form that active state?
Yeah. So the question is, do all tetramers, we can expand that to say all multimers going to be having active sites that are between these things? The answer is no. The answer is sometimes it's this way, sometimes it's not. Probably the best example is hemoglobin, where the active sites are actually really quite separate. It's where you're interacting with the oxygen atoms are in each of the hemes, so each of them in and of itself has some activity. And it turns out that it's the interactions between those different monomers are important for the regulation of each of the different ones, as opposed to being important for the actual activity. Probably one of my favorite multiple polypeptides coming together is in the RNA polymerase. And this should actually be more specifically the DNA-dependent RNA polymerase. This particular one is the yeast structure, and it's actually not quite complete. We've got here, I think, 12 different subunits labeled here in different colors associated with the DNA. Many different proteins. In fact, the active site is really just present in one of those separate polypeptides, but all of them coming together, you have to have all of them in order to get, first, the function, and second, the regulation which you get for these whole processes. And in fact, this has now become a relatively small structure, but it also emphasizes the relative size of your protein relative to the nucleic acid here. And as you remember, these ribbon diagrams aren't even showing the side chains. So if anything, it's even larger volume which is there. Mention that most of the time you have to have structure in order to have function. There are a couple of examples where actually the function is present because you have a disordered protein, so you actually don't have a stable structure. That's what's shown over here. This is best known for the nuclear pore complex. So you have proteins which are just basically all of the side chains. The backbone is just flopping around. Turns out that that's really important for the function of the nuclear pore complex. A lot of people used to think that non-structured proteins had no function, but in the last few years we've figured out that there are some. Much more common is that you have a stably non-structured protein that once something happens to it, then it becomes structured and then gives you a function. So this transition between not having a stable structure and having a stable structure is kind of a switch going from off to on. That can happen through interactions with other macromolecules, in this case, 2 different proteins. It could be some kind of modification that happens to any of the individual side chains. We'll talk a lot more about this in the next couple of minutes. Very common, that's going to be phosphorylation. Once you have phosphorylation, modification of the protein, then it has a structure, then it has a function. Or sometimes these parts of proteins that don't have very stable structures may just be kind of like a chain and holding a separate piece of your protein, say a separate domain, just close to where it needs to be. And if you think about a cell, we've got all of these different proteins floating around. And it turns out you're an enzyme that needs to make one reaction and then pass it to another enzyme that's going to have a second reaction. It's going to be a lot better if those things are close to each other rather than far apart. So evolutionarily, having this flexible domain, or in many people we'll just call this a tether, between the two parts of your protein is really quite a common process. And probably the most famous or infamous of these structured versus unstructured proteins are the so-called amyloids. Most people think about amyloids in terms of neurodegenerative diseases. So these are proteins which will have actually two very different structures. Some structures which seem to be perfectly good, some structures that seem to be really bad because they end up precipitating, making what are these called really long fibrils. And the fibril process is where you have multiple beta strands, which all then line up and give you this what's called a cross beta spline. All of these together give you a particular structure, and unfortunately, aggregates, things that form in the brains of people who have neurodegenerative diseases, or also mad cow syndrome, where you've got these different structures which are forming. But it turns out people also used to think this is just bad, neurodegenerative diseases, et cetera. It turns out there are quite a few proteins that do this on a normal cell basis, and they turn out to be important for holding other proteins together, which could be in different parts of the cell, also known as granules. What's a granule? This is something you see when you do microscopy in various cells. You'll see these subcellular structures that turn out to be very important, and we know probably the most about them for RNA recycling. So thinking about messenger RNAs, et cetera. And those are probably held in those places through these amyloid-type proteins, but are just, they can go back and forth. These arrows actually should be going in both directions. So you can have these beta spline structures that form, but you can also have these unfolded structures, which are formed relatively. Yeah?
Are these also called prions? Or
Okay, so the question here, are these also called prions? So those that end up causing disease, which are the things like mad cow, Creutzfeldt-Jakob, et cetera, those are what are known as the prions, but those are specifically the ones which are causing disease. Whereas in this case, the more general case are amyloids, which are going to have these exchanging structures. One of the things that happens with prions, which is kind of the problem, is it's usually much more of a one-directional, so it's always going from this unstructured or one particular structure to another one, which causes a disease state. Prions are amyloids, but not all amyloids are prions. But prions are defined as with the, generally with disease and disease state. Okay, last clicker question for today, I promise, is proteins that have at least blank amino acid sequence similarity identity are likely to have similar structures. Everybody's going to get this one right, right? Yes? Good? All feel happy? And go back out into the snow? Again, feel free to discuss these things. I can't force you to be in little groups, but it's what everyone wants me to do. But too much like work. I've also heard that most people don't like it. So it was not terribly popular. The question is, did it work? Okay, yes. We all agree more than 30% is the best way. And anytime you have 45 and 55, yes, they're likely to be, but the best answer here would be 35. And yes, there will unfortunately be questions like this on exams where I want the best answer, not just an answer which is correct. Okay, wanted to finish up today talking a little bit about enzymes and enzyme structures. as well as a little bit about regulation. This is basically what I've mentioned before. Most proteins, most of those extra amino acids that are out there are really just to put a couple of amino acid side chains into the right place in order to give you activity. And in the case of enzymes, that's usually generating some kind of binding site, like here, for instance, where we have side chains, serine, arginine, glutamic acid, threonine, and serine, all around where these ligands, the interactions are, two different nucleotides that are associated with each other. And what happens with most of these enzymes is they will bind to usually something that's in the middle of a chemical reaction. And so that will stimulate your chemical reaction by lowering the activation energy barrier to whatever chemical reaction is going on. And as I mentioned on Wednesday, that will really accelerate the rate of your reaction. And so if you have an energetically favorable reaction that has a high activation energy barrier, it's never going to happen. If, or at least very, very rarely, if you have a lower activation energy, it's going to happen much, much more quickly. And basically, none of biology would be possible if you didn't have these enzymes. And most of the time what they're doing is stabilizing some kind of chemical, it's called transition state, something between what's going on in your particular molecule. It's not just that interaction, like the interactions between the different domains in neuraminidase. Often you'll have other kinds of interactions with different ligands. This is what's called allosteric regulation. We'll talk a bunch about this when we talk about transcriptional regulation much later on in the term. But all that allosteric regulation is allostery means somewhere else in space. So if, for instance, you have an enzymatic reaction, you've got the active site that's going to be interacting with your transition state, then there'll be some other part of the protein that's going to interact with something else. And whatever that something else is, through this structure, is going to cause a change at your active site. And that's all that allosteric interactions mean. Something that's going to bind to your protein in one place, cause something else to change somewhere else in the protein. This can be either in a positive state, so you have what's called an allosteric activator. So here, when you have molecule X around, that allows glucose to bind to this particular enzyme, so you get lots of activity. Or you can have the opposite kind of allosteric regulator, which causes this protein to have a different structure and therefore block the kind of activation which is there. So that's nice kind of general picture. I want to spend the last few minutes talking about some very specific ones. And this has to do with signaling and various different signaling proteins. And again, it's all about changing structure. Changing structure, changing kind of interactions that are going on. There are really two different ways that this can happen. You have phosphorylation, which happens in proteins, and these so-called G proteins or GTP binding proteins. Kinases and phosphatases. Kinase, what that does is it puts a phosphate onto some other molecule. A protein kinase puts a phosphate onto a protein. I'll give you one guess what a nucleotide kinase does. Puts a phosphate, of course, onto different nucleotides. Then a phosphatase will take that phosphate off. It's this phosphate added here. One of the things with phosphates is they're large. They've got a lot of negative charge. And so that introduction of negative charge to your protein is going to change the structure. And that changing of the structure will lead to some kind of different interactions that are going on. The other major group that we'll talk about here are the so-called G proteins. Here, instead of an actual covalent modification, putting a phosphate on, it's actually the change in a bound nucleotide, here, GTP. When you have GTP bound, you have one structure. You have hydrolysis of this phosphate, and that leads to now a GDP-bound form of your protein, which has a different structure, and you have a different structure, you have a different function. No class on Monday. Hopefully, assuming that something melts, we'll see you on Wednesday next week. Stay warm, stay safe. Pardon? Yes, sorry. Thank you for the reminder. The online quiz that you will only get points if you start. Doesn't matter if you get any of the questions right, but you will get points if you do it by Monday. Yes? Am I going to post the answers to that? No.